So for those who don't know me, my name is Mike MacCoss. I'm a faculty member here at the University of Washington, and this has become kind of an annual event for us. I think this is. I hope you'll find this very engaging. The purpose of this course is definitely to help teach people some of the basics of quantitative proteomics. It's an intensive course. It definitely will be. You'll be busy. It's a long week, and we definitely. And I think I'll let you. You'll meet different people throughout the week, and people will introduce themselves during that time. But I do want to highlight a little bit about some of the kind of the expectations and et cetera in the class. So what should you basically expect in this course? And so kind of unusual, but we actually should expect kind of pretty beautiful weather here in Seattle. We've had like over 32 days, I think, of continuous sunshine, not a bit of rain, and as you can tell from all the lawns and grass around here, it's definitely unusual to have this sort of dryness. And it looks like it's going to extend for the rest of your trip here for people who are not from the Seattle area. This is the way the weather always is in Seattle. So, no, seriously. So what should you kind of expect in this course? So the first thing is I think you should expect to be challenged. The projector is a little dim, but it's unfortunately there's nothing we can do about that. But there should be material that you know, but there also should be material that we present that you don't know. There's going to be some things that some people will feel like review to them, hopefully, and then there'll be some things that people feel like. Obviously, the only purpose to take a course is that you're coming here to learn something new. One thing that's kind of key is you should expect to learn the fundamentals of targeted proteomics. It's impossible to become an expert in a week on any topic, but I think people should become fairly proficient in these ideas, the concepts, etc. And there's going to be some major thought leaders here that we're going to be presenting. One key thing that will be presented, I think, and discussed is you should learn that quantitative proteomics is not possible without extensive quality control and system suitability. There's lots of ways people can do this, but we will teach you ways that have worked for us. This won't be all-inclusive, but there will definitely be some interesting and useful use cases. You should learn when a measurement is and is not quantitative. There's a lot of things in the literature that discuss quantitative proteomics, and from my perspective at least, I would say that most of them are not quantitative. We'll talk about this, and definitely we're open to kind of discussions and kind of debate on that topic. As I said, not everybody will agree on this. As I pointed out, the fact that there is an enormous amount of literature where people present things as being quantitative, which we don't necessarily believe are, means that not everyone will agree. You should expect to be proficient in the use of Skyline for quantitative proteomics. So, if you've looked at the course schedule at all, this is you'll find that Skyline is a major component of this. It's a fantastic and very powerful piece of software, and I think you'll again, while you can't necessarily be a complete expert, you'll learn, I think, a lot of tricks and key components and ability to make use of it for your research. So this is another thing that may sound a little strange. You may hear contradictory things from different instructors. And also I'll point out that I'm pretty sure what will happen is different instructors might present the same things different ways. Proteomics in general is still a fairly new field. We're still trying to wrap our heads around what's the best way to do things, and we don't always agree. And I think that's actually not a bad thing. I think as long as people are open to kind of that debate and that discussion. So I think this is kind of a key component. So don't be surprised if you hear things where somebody else contradicted it previously. And the other kind of component here is you should expect to have fun. Right? This is we were sitting in this classroom for basically a week long straight. Well, it's beautiful weather outside, but I hope that people enjoy this course. And the goal here really is to kind of learn some things, but at the same time to meet new colleagues and to have fun. So here's a couple things a little bit about our expectations. We expect you to challenge us. You won't hurt our feelings if you disagree with us. We're happy to hear about different things, different perspectives, things that work for you, but maybe presented differently within this course. We definitely want feedback, and we also want to hear that if you want more. In fact, one of the things that Brendan does in the mornings is these sky jams, and there is really kind of an opportunity to kind of dig deep and to learn a lot more about some of these tools. And we definitely expect you to kind of make use of these opportunities. We expect different course participants to come from very different backgrounds. In fact, we've known that some people come from small molecule backgrounds trying to get into proteomics. Some people are very new to proteomics just in general. Some people have been working in proteomics for a very long time but wanted to learn something about Skyline or some other aspect of this course. But the other thing that we've learned is that we've come to expect that we often learn from you guys a fair amount. You guys are very focused on very specific biological questions. You have certain needs that you have to fulfill, either in your own personal research or offering resources to a core facility. And in that process, we often learn a lot in the process by thinking about what you guys need. This is a key component. If you sit here largely quiet, you won't get as much out of the course as if you participate more. So don't hesitate to ask a question. If you have a question, it's almost certain that somebody else has it too. And it's good to kind of bring these questions up early and often, and it definitely won't distract on things. So we definitely encourage that. We also encourage you to kind of, during coffee breaks and during some of the other social events that we have, to interact with your colleagues and also the instructors, because I think this is a great opportunity for people to kind of meet. What we expect is you will hear things that contradict the literature. We've had plenty of times where we presented things and someone says, well, I have this paper here that says the opposite. That's not necessarily a bad thing, as I pointed out before. And I think that's, again, it's kind of a growing field, and this is part of the growing pains, I think, in a growing field. And again, you will hear differences in opinion from different speakers during the course, and the people who are presenting here, we've all worked together for quite a while. We definitely know about each other's differences and opinions, and in many ways, we've actually kind of cherish and kind of look forward to the fact that people have differences in opinions, and we don't want to necessarily present one opinion and one viewpoint. Okay, so what I'm going to do a little bit is to kind of get people into a little bit on the same page, and a little bit of introduction to quantitative analysis of proteins, and this may or may not be reviewed to certain people, and I'm sure regardless of whether or not this is new for people or not, it's probably presented in a slightly different way than what you've heard previously, so I think it's all kind of good, even if it is a review. So one thing that I've always found pretty exciting, of course, is why is it that we are doing proteomics? Our lab sits in a genomics department. We get asked this all the time. Why aren't we doing transcript profiling for everything? It's so much easier. You just get sequencing reads, and it becomes very digital, as often people will often say. And I think this figure is not the only figure, but it's one figure that kind of highlights it fairly well. This is from Mike Hengartner's lab in 2009. And it's actually kind of an interesting observation in the fact that what they did is a fairly straightforward experiment. They took C. elegans and they took Drosophila, which are 100 million years diverged, and in that process they measured protein levels between the two, and they measured transcript levels by two different methods, either Affymetrix and SAGE, a serial analysis of gene expression. This is before RNA-seq was widely used, and the other kind of key component here is that they took a clear one-to-one ortholog, where it was obvious that there was a protein that's in one, that's the clear single ortholog in the other species. It turns out that if you look at the protein levels, there's a really good correlation between protein levels from where there's a clear one-to-one ortholog. But if they look between the protein and the transcript, that correlation is pretty poor. And also, if they measure the transcript levels by two different methods, the correlation's poor. And if you look at the correlation of the transcripts between species, it's also particularly poor. The authors speculate on this, and Christine Vogel's also done a lot associated with this as well. And this is and the idea here is that and the rationale here is that the selective pressure is acting on the protein and not the transcript. There's no reason to keep a transcript at a given level, and there's many ways in which you can alter the level of the protein, not just or the activity of the protein. So if you have a protein that's conserved over 100 million years, the fact that it's still highly correlated with its abundance in a completely different species that diverse is pretty remarkable. So another common question that we get asked and that other people often bring up is why use mass spectrometry to measure proteins? Well, there's a lot of problems with basically immunoassays. There used to be this basic idea that mass spectrometry would be used to get candidates, and all the validation would eventually be done by immunoassays, or at least clinical immunoassays in the clinical lab. But there are problems. In general, immunoassays are singleplex. There are cases where people multiplex them. They don't work very well in general. But so in general, when you think about immunoassays or affinity assays, they're singleplex. There are poor standardization, so whether or not you use one platform or another, or one set of antibodies or second set of antibodies, people tend to get different results. There's this artifact known as the hook effect, which is that as you get to higher and higher abundances, you actually get... Hey, Nat, it looks like the pointer doesn't have batteries. Is that... or... Listen, yeah.
I guess
Okay. No problem.
Okay.
Well, maybe I did turn it on. Oh, I should have turned it on. That would have helped.
Yeah.
Okay. So then there's the hook effect, which is where basically if you start to measure, if you have a really high abundance of an analyte, you start to get to the point where both your two portions of your sandwiches actually both recognize the analyte individually, and they don't actually form a sandwich. And so you actually get a false negative measurement or an underestimation of your level. And then the other problem, which people don't think about too often, is just the presence of autoantibodies. So in most cases, a lot of proteins generate an immune response. A lot of plasma proteins do. And so you actually have in your own plasma or any other animal, but to be honest, actually is often that these proteins are coded in autoantibodies. So it's really hard for then another antibody to recognize that protein. But if you go about the process of digesting proteins and things, it's fine. So mass spectrometry is definitely a potential solution. So the first aspect is it's direct detection of the analyte by mass spectrometry. If you go about doing this correctly, you could actually have absolute structural specificity. There's this. It's been demonstrated that we can, using the same standard, to have inter-laboratory calibration. So basically, a quantity measured in one lab will give the same quantity measured in another lab. There's definitely improved specificity. Tandem mass spectrometry is definitely offering very incredible specificity. And there's no sandwiches formed, so you have no effective the hook effect. You do sometimes get saturation of the signal, but that's a slightly different problem. And you get around the autoantibody problems by destroying the proteins by digesting with an enzyme. So if you think about quantitative analysis, just in general, this isn't limited to mass spectrometry, is you tend to have some response, so some measured intensity with some amount that's being measured. And regardless of whether or not this is a BCA protein assay, whether or not it's some assay to measure RNA content or some fluorescence assay, you generally have some measured intensity and it correlates with the measured amount. And all assays, regardless of what the dynamic range, have some sort of S shape to them, where you have a region that should be linear, so the response is linear. You have a lower limit of quantitation and an upper limit of quantitation. And then, of course, below that lower limit of quantitation is the minimal range in which you get the detection, which we call limited detection. And these are kind of a critical kind of component, right? So this is what often people call a calibration curve. So you have known standards that's used to often assess this. You assess the range over which a response is linear. There's a range in which the detector begins to saturate on some instruments, and there's a level in which you can keep getting dilution of the material, but you don't necessarily get a decrease in the amount of signal. People often talk about using an internal standard in mass spectrometry. We're particularly lucky that we're making measurements based on mass, so we can use a stable isotope labeled internal standard in many cases. But the problem is identical. You basically have some quantity or amount material, and the only thing that's changed now is that the intensity isn't just the raw peak area, etc. It's now a normalized intensity. And it doesn't matter whether or not you normalize it to a stable isotope labeled internal standard or just a common peptide in the sample. It's just basically some normalization strategy. Sometimes people will even normalize to the total ion current, etc. So when you think about this, if you just have this case of measuring some analyte with massive charge M, and you have these isotope distributions, in almost everything that we'll be talking about in this course, you then go about taking this and converting it to extracted ion chromatograms. So you have time being plotted on the x-axis and intensity. You have what we call an extracted ion chromatograph with an area A, and we make some assumption that this area A is proportional to the moles of the sample. And so if you have a calibration curve where you have some analytes of known amount and you have some measured signal, you should be able to convert that measured signal to a quantity using just basically a simple linear equation, etc. And people will often discuss a lot about requiring the need of stable isotope labeled internal standards. You don't need a stable isotope labeled internal standard. You basically just need analytes of known quantity. And the problem here is what's known, because in general we're sort of having to rely on some sort of standard where it really isn't a known quantity. In many cases, when you actually do this in practice, while you may measure many different points during the validation of your assay, when you're actually in practice performing the experiment, it's hard to see here, but there's people will often measure just a couple points. And I know that people in our lab and Andy Hufnagel will probably present this idea of using, in many cases, a single point calibrator. And this is actually the same idea, but you have to make some assumptions about the intercept. But in general, you can get by often with just a single point calibrator. So things don't change at all, really, when you use a stable isotope labeled internal standard. You now have this component of mass and this mass plus number of neutrons that are added. And then you now have two extracted ion chromatograms. And you have now an area ratio, which we believe is proportional to the mole ratio. And you can then use this, again, based on, we have this measured signal, which is now a normalized signal. It's an area ratio with this known amount. And, of course, we can rearrange this linear equation, of course, to solve for the known amount using standards. So what does it mean if these are your 2 points? So let's say you have a case where you have a healthy and a disease. You're measuring 2 quantities. And you may or may not have done a calibration curve, but these are the 2 points you're measured. One's a low signal, one's a high signal. And what would this mean? The 2 yellow points. Yeah, so there's you believe there's a difference in abundance, right? Okay. What else does it mean?
One is quantifiable, one is not.
Yeah, or if you were trying to measure a ratio between the two, they may not necessarily reflect the amount of moles, right? The actual amount of quantity. Do we think this happens a lot in proteomics? Yeah. Right. Okay. And so, so does that mean that so for something to be quantitative, the change in signal should be reflective of the quantity? So in this case, the change in signal, the measured intensity, will be an underestimate of the quantity, right? And so often people talk about this as being kind of signal compression, but in general it just means that one of the points, or multiple of the points maybe, below the limit of quant, so we don't get a linear response. Right, so this is partially why I said that I believe a lot of the data that's presented as quantitative proteomics may or may not be quantitative, because while this may be differential, so these things may be a statistical difference, you could say that this thing is more abundant than this one. You may not say if there was a tenfold difference in signal intensity that it may be greater than tenfold difference in the quantity. In many cases, people are happy with that. So that's not too bad of a ding. It's just things if you were trying to expect to say you wanted to know the actual quantity differences, that's an important component. So if let's say you wanted to figure out whether or not you are in this linear range, generally people make calibration curves. They make dilutions and demonstrate when they make a dilution that their change in their signal response is appropriate. So I want you to take just a couple minutes and just kind of think about this for a second. So let's say someone came to you with 50 peptides and said, look, I need you to design an experiment where you can actually determine whether or not the change in response for these 50 things that are in your complex matrix really are going to be quantitative, meaning that they're in the linear portion of the response range. And then make another assumption that someone's going to come to you with greater than 1,000 peptides, basically a proteomics experiment with lots and lots of things. How would you confirm that those things are linear? Just take a couple minutes and then and think about how you might do this. Would you take different approaches? See someone nodding their head? Yeah? So if you had 50 peptides, what would you do? Or we could even just say 5.
Preferably, I'd do a statistical limit of detection on it, and a limit of quantitation.
But so on what?
Standards.
On standards, right? So you purchase standards. So you could potentially purchase 50 standards at maybe $400 a pop. That's still a lot of money, but it's doable. But you definitely couldn't do that really for 1,000 peptides, right? So so if you had standards, so basically you'd have 50 peptides, and you would make dilution curves of those 50 peptides, and you would then use a statistical approach to see whether or not the response is changing appropriately, and whether or not you're within a linear range. And people will be discussing later this week on different ways in which we can assess that in limit of quant. So what happens is the response curve going to be the same in a buffer versus the matrix? No. Okay. All right. So if you have a matrix of which your peptide may be in it, how do we do that? You compare it with the background proteins. Yes, you can maybe do sometimes what people call method of standard addition. So they look up, look to make sure that the response goes up from that appropriately.
Or use the isotope labeled.
Yeah, sometimes people use stable isotope labeled and they confirm it using that the response is linear with the stable isotope labeled analog. But that's a little different, right? Because the isotope labeled one may or may not respond exactly the same as the unlabeled. But still, so basically what we're thinking about is that we have standards that we purchase. Now let's say someone wants to do a swath experiment or a DIA experiment or a max quant experiment. And you need to be able to say whether or not these 1,000 peptides are quantitative. How would we do it? Mixed proteomes. Yeah. So I think that's basically one of the approaches that we definitely take. So one idea here is that we have a pooled reference. You have a mixture. You have a sample, and you make. And one of the things that you can do is you can mix them in two different ways. And basically, you're making a dilution of the entire proteome, but you're trying to keep the complexity of the mixture the same. And so in a case like plasma, we often use chicken plasma because it's fully diverged, so none of the peptides or at least very few of the peptides are conserved. In a case of something like a human cell lysate, we can use a SILAC labeled analog of it and make a dilution of it. But basically the idea is that you're taking every single peptide and making a dilution of it, but we're adding back a balance, right? So basically we're adding back something with a complex matrix to make sure that there. So this will be discussed actually a fair amount later in the course, but I think it kind of gives you a sense of a possibility, right? So just gives you another way of looking at it. Here's showing 15N yeast being used as a diluent of this unlabeled yeast. And basically in this case, we demonstrate that across the entire range of the dilution, we basically have a linear response. This protein right here, we end up having some plateau occurring down here. And because in this strain of yeast, we happen to know a good estimate of the copies per cell, we can then get our lower limit of Quant actually done fairly well using this basic estimate. So this is another thing. This is something that Andy Hoofnagel often advocates. So if you're trying to measure one protein or condition, he will often take, again, his two most extreme samples, so the one with the lowest signal and the one with the highest signal, and he then starts to make dilution ratios of them. And while this doesn't let you get the lower limit of Quant, it allows him to determine whether or not the response is linear between them. So if he has the extreme of the lowest level sample and the highest level sample, and he starts making mixtures of them between them, he knows that he has to have a linear response between them, right? And if there is no change between his disease and his healthy, he's fine too, right? And he says, okay, it's all set, there's no change, so I don't need to worry about whether or not that change is quantitative. If the protein he's interested in is changing, he's taken his most extreme disease case as undiseased case, he then starts to, he has to confirm whether or not the response is linear between the extremes. So this already got brought up a little bit too, but the other way you can basically do this is to essentially take method of standard addition. And if you actually have this is a common way in which you can assess whether or not your response is quantitative. And that is, you basically spike in a given known amount, and you demonstrate that the quantity of the increase in the amount of a spike actually goes up appropriately. You can't use this to assess the limit of quantitation, but you can use it to assess whether or not the sample pre the spike is above the lower limit of quantitation, because this line won't go through that point otherwise. Okay, so here's some other things to consider, and these are kind of these are some of them. They're in many ways they kind of ding proteomics a little bit, or demonstrate that it's a little bit more complicated. So the first thing that it's important to kind of point out, and this is something that I know that our lab struggles with a lot, and I think other labs may struggle with this too. People will often say, well, I want to know the protein changes. I don't really care about the peptide changes. Well, you have to kind of think back a little bit about what does it really mean, right? So I've kind of drawn a protein that we've kind of stretched out. It looks kind of like a worm, but that gene is translated to a form, and that, of course, that protein, is modified. And in many cases, you'll have 20 or 30 proteoforms for each gene. In fact, there's been cases where people used to show with two-dimensional gels where someone cut out 100 spots on one gel and all of them map back to the same gene. And those are true, real cases, right? So now if you measure, and they're all at different levels, and if you measure one peptide, why do you think that that peptide could give rise to that information about an individual protein, right? And so we know that proteoforms exist. We know that these things all exist in different amounts and different quantities. And if you actually measure different peptides that span them, they often change in different directions. And those are real measurements, and they're important measurements. And you'll hear about other cases where people get different peptides moving different directions. So in general, people will start with thinking about, okay, I need to build an assay for a given gene product. And that assay will focus on which peptides respond well, et cetera. And those are all valid and perfectly important things to keep in mind. But it's really important for people to keep in mind that there's this underlying thing going on. And that you have a gene that is producing a protein, and those proteins are being modified extensively. It could be 20 to 100 different proteoforms for every different gene, all at very different levels. And these things then complicate, of course, these analyses. So it's one of those cases where it's really hard to put Humpty Dumpty back together again when you don't know all the information to start with. The other thing that's kind of important, and this will be presented quite a bit by Sandy Spencer from our lab, and that is that peptide intensity varies over the course of digestion. We often think about taking a protein, you digesting them, and some proteins, you know, they kind of go up and they plateau fairly easily in a fairly short period of time during the digestion. Some peptides go up and then come back down again. Other ones just don't ever seem to really plateau, etc. It depends on which peptide that you measure. Some peptides may respond better than other ones, but these ones then definitely seem to have kind of a leveling stability. So if you have a digestion time course that kind of sits in this vertical range of the digestion period, this is something that's kind of complicating, right? If you have a peptide that kind of goes up and then comes back down again over intensity, that makes it complicated to kind of refer back to that quantity to the underlying protein levels. Again, these are just challenges, but they're very important for people to think about when they're trying to build a quantitative assay. So people often talk about having stable isotope labeled internal standards, and it's important. So let's say you have this peptide sequence here, and it's got 15 nitrogens in it, and let's say you happen to be able to purchase 100% atom percent excess 15N. Right? So I'll point out you can't buy 100% 15N. It's more like 98 or 99%. And if, let's say, you happen to have some oracle told you that you really mix them perfectly 1-to-1, right? Would you get a 1-to-1 response? It's kind of a trick question. Yeah, we're assuming that they elute exactly at the same time, so that any suppression would occur equally between them. The only thing that differs is this 15 extra neutrons. Let's say also there's no signal suppression. Okay, let's say the ionase perfectly together. Would you expect to see a 1-to-1 response? Pardon me?
Expect to see a 1-to-1, but probably you won't.
And so and there was a question about bias and things too. So let's say, okay, let's say there is no bias in the quad. Let's say everything is going perfect. So the key is no. Theory would say there would not be. The 15N monoisotopic mass would be a little bit more intense. Does anyone know why? Because there's 15 less 15Ns contributing to the M+1 isotope. And nitrogen is like 0.35 percent 15N by natural abundance. And so if you now have something that's 100% enriched in 15N, it means this isotope distribution is slightly skewed. It's got 15 extra 15Ns, but now this one has 15 less nitrogens contributing to the M+1 isotope and the M+2 isotope. So the isotope distribution is skewed, and the monoisotopic mass of that labeled isotopomer is a greater proportion of the total isotope distribution. And it gets, of course, it's more skewed as you would predict with 98%, which is what you normally purchase. You have then a fairly large contribution to the 14N and to the 13 15Ns. And of course, when you get down to lower levels, there's a problem. Now, if you're just using this as an internal standard to normalize your signal, to correct for ion suppression or quadrupole bias or autosample injection issues, etc., there's no problem. But if you're trying to use this to calibrate your signal response, it's just something that's important to kind of keep in mind, right? Now, I'll point out that 15N is like the best case scenarios. A lot of people say, well, I don't use 15N. I'm going to use 13C because that's what I see all these SILAC papers out, etc. 13C is probably the worst contributor, right? So let's say you had the cysteine that's labeled here with an ICAT. This is a reagent that people don't use it often anymore, but it's satisfies the example. So this peptide is labeled with 9 13Cs at greater than 99% atom percent excess. And if you mix them at a perfect 1-to-1 ratio, and you were able to measure them perfectly with no error or no noise, the monoisotopic masses would be off by about 10%. And that's because the contribution of these different isotope distributions are very different. And so this, again, isn't that big of a deal, right? Because we can calculate these things, we can adjust the things of things, but knowing what this enrichment now then becomes very important, right? And so a lot of the things that we'll talk about here throughout the course and Andy Hoofnagel will spend a lot of time thinking about this too, is whether or not even using a stable isotope labeled peptide is a good thing to use as a calibrant. It's maybe a good internal standard, but may not be a good calibrant. Anyway, and there'll be lots of discussions about, ways in which you can calibrate your signal response. I'll just point out here that actually, deuterium actually isn't too bad because, again, it's like 15N. There's a small natural abundance contribution. Now, there are cases where people bring up, rightfully so, cases where chromatographic shift, et cetera, and isotope effects. But in general, the effect on the signal intensity is fairly small. So I just want to bring up a couple points about study design. This will get talked about a lot more, especially when Olga Videk is here on Wednesday. But it's important to kind of keep just to think about this a little bit. So one thing I'll point out is a lot of people in proteomics, my lab included, and I know that a lot of the labs that we work with also probably have the same issues, probably don't spend enough time thinking about our study design as thoroughly as we should. So one is the thing about is how is the experiment performed? What add unnecessary variance? Are we eliminating or minimizing systematic effects? Is your study powered correctly? The idea and concept of pooling or not pooling samples. If you pool, are your results what you think they are? Are you using the right statistical tests? Are you correcting for multiple hypothesis testing? So these are, the bottom one is particularly important for proteomics, right? Because if you have 10,000 peptides and you set a p-value of 0.01, right? You're going to have a lot of things changing just by random chance, right? That because you're measuring so many things. And people will often say, well, I want to measure everything. I want to measure as many things as possible. And what often happens then is some of the things that they're really interested in now go out to the point where they no longer have any statistical power to find those things because they are measuring it in the context of everything else. This is kind of just an important thing to keep in mind. So if you don't know about a lot of these questions, it's not unusual. A lot of people, and especially I, haven't thought too much about them originally. We've spent more time thinking about them more recently. But one thing I will kind of bring up, and that is to involve a statistician early. One of our first real mistakes about this was we did this enormous proteomics experiment. And I started to sit down to try to figure out how we were going to analyze the data. And in our lab right next door to us was John Story. People may know him. He was the person who the Story Tips-Shirani. He invented the Q-value idea for correcting multiple hypothesis testing. And I went and talked to him for a while, and he'd sit there and he listened to me for quite a bit of time, and he sat there and said, why, oh, why didn't you come talk to me before you did the experiment? And so I think that's kind of the key thing. If you're not going to be the person analyzing your data or doing the statistical analysis, you want to make sure that you're involving the person who is going to be helping you early on. In fact, I think that's really kind of critical. And also, I think it's really critical that you build kind of a working relationship with the statisticians that you've ended up interacting with. They actually do need to understand the data. And I think the more people we can kind of help understand the data, the better off we all are. And I think once you have those people who, it becomes painful initially trying to get them to understand the data, but once they eventually do get it, they become lifelong colleagues. So a couple things to kind of keep in mind, and that is that experimental variance is unavoidable. And so what do? So we assess it and account for it. There's no point ignoring this. And so, again, you have to have replicates. You have to have the ability to assess the variance. If there is an added step of unnecessary variance, you should go through the experiment to figure out that contribution to it. if there's a kind of a take-home message, and that is the variance is almost always larger for biological variances versus technical variances. People will often say, well, how many technical replicates should I do? We'll often joke and say, do none and go get more biological samples. Because in the end, the variance of the technical replicates is very minimal compared to the biological replicates. And you need to basically, in order, you'll gain more power by actually having more biological replicates than you would by doing more technical replicates. I'm not saying that technical replicates are useless. I'm just saying that in the context of the cost of doing an experiment, they end up being small relative to, the value tends to be small relative to that of adding more biological replicates. So this was in this review paper from Catherine Lilly a long time ago, and she talked about common sorts of schemes that people will often use to kind of find differences. So let me kind of give you a little bit of her nomenclature here. So she's got, of course, A, B, C, and D are sort of 4 different examples. In A, there's this control and the treated. And there's a biological sample as B as biological samples. T are technical replicates. And one thing that she points out, and I'll point out as well, is that, again, there is one biological replicate and one biological replicate. This data is useless, basically. You can't determine whether or not the biological change is different from either just 2 individuals that were controls or treated, because you have no idea what the variance is between the biological conditions, right? So if you were to take everybody in this room who's a vegetarian or those who's not, right, and if you took just any one person, you really couldn't determine anything from it, right? So and so basically, you really need to be able to have multiple different biological replicates to assess that biological variance, which is going to be much greater than that of the technical variance. And do you know what? I bet if you actually opened up like the most recent, JPR or MCP paper, there'd probably be lots and lots of experiments that do things like this. I'm just highlighting the fact that these things still are an issue and a problem. Yeah?
How do you define the technical replicates here? Is this the reinjection of the samples or the same cell lines? How do you define that?
So in my case, it could be any of them. So the technical variance would basically be if you took the same biological sample, let's go, for example, plasma or tissue biopsy, and you took that sample and you prepped it multiple different times, or you made multiple different injections, regardless, it's still going to have less variance than if you took 2 different individuals or 3 different individuals, right? In general, our sample handling artifacts, which tend to be greater than our measurement artifacts, but in general, the biological noise is still greater than that of the sample handling errors and also the mass spec measurement errors. And also, if you have multiple different biological replicates and they're all prepped separately, those take into account the technical variant. So this is definitely a much better study design, where instead of having 3 technical replicates for 1 biological replicate, you have just 3 biological replicates. That may not have enough power, but still, it's definitely a much better design than that of just using different technical replicates. Then there's this type of experiment here, which is why we kind of illustrated the challenges of doing technical replicates. You kind of get this explosion if you do biological replicates and then add many different technical replicates. In general, we have this case of a mass spectrometer costs you X amount of hundreds of thousands of dollars, and let's say you depreciate it over 5 to 7 years or something like that, and you start to work out then what the cost is per hour of operating it. You're basically having now the cost of each biological sample going up 3x in order to measure it. And that's why we often say, yes, it'd be better to have more biological replicates. You often have collaborators that say, well, the cost of actually getting those extra samples are expensive, but then it becomes this case of where you have to do this cost benefit, and is it worth doing an experiment that adds a lot more mass spectrometry time or personnel time to prep the samples versus having a case where you just obtain more biological replicates. So then there are other schemes too, where you have like basically cases where you pool some subsets of the samples, but you're still able to derive some of the variance between it. I would definitely say if you use some pooling scheme, it would definitely be in your best interest to kind of consult with a statistician beforehand. You definitely need to be careful with pooling of samples because while, yes, you can account for some of the variance, the biological variance, your results may not necessarily be exactly what you think they are. There could be some significant outliers that may not be reflected actually in the data this way. So basically the technical and the biological variance is decoupled, and biological variance is almost always greater than technical variance, so error on biological replicates whenever possible. I won't go into too much detail about this, but one thing is that is to think about your power, and sometimes this may require the need to do a pilot experiment first. You need to know a little bit about the variance for your individual peptide that you're interested in, or the individual protein that you're interested in. And so basically, as you increase the number of replicates, your power increases. So that for a given significance level, the number of things that are changing in the sample for a given effect size, increases. So your ability to find those differences and find statistically that they are different, the means are different between those groups, it improves. One kind of rule of thumb that we've kind of learned, at least in our lab from doing a lot of power analyses, is that if you take a multiplier of about 5 or 6, it definitely helps improve things for the multiple testing correction. So one of the things that this is only showing here is a case of any one analyte that's being measured. But if you're measuring 10,000 analytes, this gets much worse. You lose a lot of power again from that. But if by doing a proteomics experiment, you should basically multiply your number of replicates for each condition by about 5 or 6. So if you needed 3 replicates previously in order to find something being significant for a single analyte, like a targeted analysis of one peptide, you basically need 15 to 18 per group to measure in the context of 10,000 peptides. Now, there's a lot of great analyses that were done with this in the kind of the GWAS field, where if you're testing for any one single nucleotide polymorphism versus doing genome-wide studies, but basically the multiplier is on the order of about 5 or 6 for large-scale studies. So if something for a single peptide was significant at 3, with 3 replicates, then it should be okay in the context of a proteomics experiment with 15 or 18. If it took you 5, then you basically need 25 or 30 replicates, right, to do it in the context of a proteomics experiment. So that's something to kind of keep in mind. People like this idea of doing, MS1 profiling of lots and lots and lots of things, or a DIA swath experiment. But do you have the ability to do that 5 or 6x multiplication of the number of samples? Again, all I'll point out here is that human biological variance is a mess, and that you basically need, in order to find things that are significant, you need lots of samples. That's the only thing I'll cover with that. Okay, so another kind of component here is that there's a lot of challenges with discovery proteomics, and there's some possible alternatives. So I think most people here realize that in bottom-up proteomics, we basically take protein mixture, we digest them with an enzyme, we separate it by some sort of microcapillary chromatography, we acquire a mass spectrum, and then we use a process called data-dependent acquisition to often trigger MS2 events. And the key component here is that these mixtures are very complicated, right? So this figure actually doesn't do it justice in the screen, but this is plotted mass to charge versus time, and every basic dot here is a peptide essentially, and there's thousands of spots in this plot. And if you basically go in and look at a single, unit resolution chromatogram, there are XIC, there are hundreds, if not thousands, of peaks that have the same mass, same nominal mass. And if you take a slice of the data at only one point in time in a mass spectrum, you have hundreds, if not thousands, of things that elute at the same time but may have different masses. So again, the traditional way in which people have done this is data-dependent acquisition, and you basically sample things in real time on the fly, and this collects these scattering of MS spectra. This is a plot that Han Yin Yang in our lab had made to kind of assess how well we were potentially doing. And this is, we had this the software that basically goes through and assesses what are all the features that can be found in a given data set at a given scan number. And this is for Saccharomyces cerevisiae. It's much worse for vertebrate samples. But basically, there are times when you have over 200 molecular species at any one time. And this makes basically sampling things really, really, really difficult in order to do this on the fly in real time to sample these things routinely. So to get every precursor using DDA, we would have to collect 121 MS-MS spectra on average per chromatographic peak. And this is beginning to get a little bit better with really fast scanning instruments, but still, this is a problem, at least to make sure that you sample the same things over and over again. So the way I always kind of think about it is this old I Love Lucy episode where basically these chocolates are coming off the conveyor belt and they were supposed to not let any of the chocolates go by without wrapping them in this paper and of course, it gets to the point where they end up stuffing their mouths with the chocolates and they start, end up, putting them down their shirt and hiding them because they were supposed to be docked for pay for every chocolate that went by in the conveyor belt. So imagine being docked for every peptide that makes it through the instrument without doing MS-MS on it. Here's just again showing it another way, but basically, there's a lot of things that can be seen, and only a portion gets sampled. And I'll also point out too, and this is kind of a little bit of a controversial topic at times, is that we know that MS-MS is so much more sensitive than using MS1, but at the same time we're still using the MS1 in order to tell us what to sample for doing MS-MS, and this definitely just reiterates some of this. And if you look at it here, and here's all the features that are in basically three-dimensional space, and if you remove the ones that got sampled for MS-MS, you remove predominantly the most intense ones. There still are some medium height ones that are still not sampled, largely because they came out in the elution profile at the exact same time as things that were much more intense. So again, do we really want to be focusing most of our instrument time sampling the most abundant molecular species? An interesting thing here is that the density of the low abundance things out here at different retention times gets a lot less just because of regions in which things get sampled. So you do have these weird sampling artifacts, and you don't necessarily sample just the most intense things. Sometimes you sample some of the lower intense things just because of the hydrophobicity and the complexities of the sample. So this is kind of an analogy I like to give. This is one that Nate Yates used to always kind of hint at one point, and that is kind of imagine going to visit a foreign city. I'll give an example of Kyoto, and you're told that you can only visit and experience that city by going to the tallest structures first. Okay, so you go into Kyoto and you go to the tallest building, and it's probably like any other Western building, and you have maybe like a hotel or an office building. And it looks very similar to that of any of our other office buildings that we have here or in Europe. And you look out the window and you kind of get a good sense. Okay, well, it's a city. It's surrounded by hills. It's very picturesque. It's got this really high-speed train that goes through the middle. This you may notice, wow, they drive on the left-hand side of the road. That's a little unusual. That's not the same as what we have here in the United States. But if you look generally, though, throughout the city, it would look fairly similar. And then you go to the second highest building, and you go, okay, well, again, it looks like a hotel or an office building and things. And you go to the third most tallest building, I think. And, but you'd miss all these things at street level, right? So if anyone's ever been to Kyoto, it's just got these incredible shrines and temples and it's just culturally, like a lot of places in Japan, are very different than what you often see in Western cities. And the way that people dress is different. The types of food that people eat, can be different, right? And that's often a way that you experience foreign cities and cultures is going out to eat. And so one of the things I kind of want to point out is that if you wouldn't learn much about Kyoto by going to the tallest structures, why would we ever use this approach to understand protein mixtures? And I think this becomes kind of an important thing to kind of iterate through this course. You know, people are so focused on getting this big list of things that they don't think that maybe that the approach that they're taking from the start may not be the ideal way of performing the experiments. So what do we normally do? Well, we often build on other people's knowledge, right? So you'd go and you do a Google search and you'd say, look, what are the key places to visit? And I'm not saying that if you go and say Google, you know, the key places to visit in a foreign city, that you'll get all the best things to do if you had lived there for your entire life. But you would basically do better off than just going and starting from the tallest structures first, right? And basically, or randomly selecting things on a different street corner, right, to go visit and see. You would know that, you know, you should go see certain temples or shrines or aspects of the city or the things that are important about the local culture, etc., that you should visit. I'm sure that if you Googled, you know, the best things to go see in Seattle, you'd get normally, you know, all the regular tourist things that people would go see. You know, you'd go to the space, you know, go to Pike's Place Market, you'd go. You know, and so there would be a lot of components. You learn about what sort of foods. The seafood's fantastic here, right? And so you learn a lot about the details to actually do than just randomly going to different neighborhoods in the city. So I want to bring this back another way. So when I first started my lab, we decided that we were going to use C. elegans as an in vivo model system for proteomics. And the reason why we decided to do this was there were a lot of people here at the University of Washington with a lot of experience in C. elegans. Also at the time, sort of about 2004, it was the only multicellular organism with a genome sequence to completeness. I actually believe there's only one other organism sequence to completeness. I know we hear about all these things, these new genomes being sequenced all the time, but I'm talking about telomere to telomere for all chromosomes without any gaps. And C. elegans was the first multicellular organism that was sequenced to completeness, and now I still believe there's only one other that's been sequenced to completeness. It actually was an incredible effort that was put forward by John Solston and Bob Waterston. John Solston also worked out this complete lineage for all 959 somatic cells. So if you're thinking about trying to understand an organism to completeness, there was a lot that we already knew to completeness. We had a genome sequence. We knew all the complete lineage of all the somatic cells, but we knew very little about the proteins. There's a lot of proteins that are conserved in the pathway between humans and worms. We had proposed that we're going to use it for insulin signaling, and again, this is a figure I had in the original grant and showing, you know, look, you know, we have an insulin receptor, we have, you know, 4-cat transcription factor, we have AKT1 and 2, and, you know, PDK1, the PI3 kinase, etc. So it was a good model system potentially for studying insulin signaling. So we decided that we were just going to do a discovery approach for this, right? So we took worms, we figured, okay, we're decent protein biochemists. We'll do some basic fractionation based on physiochemical properties on the protein level, and then we'll go and digest them, and then we'll run multidimensional chromatography. So this was, you know, 30 days of basically of instrument time. Each of these things were LC-LC runs of about, you know, 24 hours apiece. And we identified lots and lots of peptides and lots of proteins. And although this was still, you know, older instrumentation, we could definitely get better now. But this is still pretty good for given the effort and the technology we had. The key thing was that 0 proteins were identified that are known to be involved in the insulin IGF-1 signaling problem. So this is kind of like going to a foreign city where you want to be a tourist. And you know what the top 10 most important things are that you want to go visit, but you go stuck and you're going to the tallest structures first to the point where you miss all the things that people have ever said that have been important about that city. So this then got us thinking a lot about using targeted proteomics. And so there's lots of different ways of doing this, but, you know, I'll start initially with some selective reaction or parallel reaction monitoring are two kind of components. And the only thing that really differentiates between the two is that in one case you measure all the product ions individually, and in another case you measure them all at once. And so in the case of a triple quad, you basically have a first quad. This will get discussed much more later today. But you have first quad set to pass just one mass that gets subjected to collisions, and then you measure just an individual mass again later. And this is why it's called selected reaction monitoring, because you only ever measure a single or selected reaction at a time. In fact, I find that multiple reaction monitoring kind of gets confusing because you're not measuring multiple reactions at a time. You're measuring them selected reactions in series, right? So the way that this data looks for people who haven't been used to doing targeted proteomics is we're basically kind of not looking at spectra anymore. Everything becomes a chromatogram. So you have this precursor ion, this product ion, and they show that a very selective chromatogram. Whoops. Where this is the Y2 ion, this is the precursor of the Y2 ion, the precursor of the Y3 ion, the precursor of the Y4 ion, etc. They all colute. That suggests that they belong to the same molecular species that has the same hydrophobicity and the same precursor information, but different product ions. The fact that they all colute basically allows us to reconstruct most of the sequence information, and this becomes a very selective measurement. So again, going back to this C. elegans project, here's the fork-head transcription factor. Here's AKT1. Here's the chromatograms now measured in 30 minutes of instrument time. And we use standards to confirm the identity, the relative ion ratios, the retention time, etc. And again, so we went from 30 days of instrument time to not being able to detect and measure these things to 30 minutes of instrument time to being able to detect and measure them. One kind of key component of this is this unparalleled dynamic range. So you take a peptide and a complex matrix. This peptide just responds really well, but you can go down to this bottom and kind of blow up this calibration curve. And you basically have 4 logs of linear response. Here's the lower limit of quantitation for this from unfractionated plasma. Now try to do that with like a Western blot. I mean, Western blot, you're lucky if you've got like factor of 10 dynamic range. So the other kind of key components to doing MSMS is that there may be lots of things that have a given retention time. There may be things that have the same retention time and the same mass. But what's the likelihood of having things that have the same retention time, the same mass, and the same product ions, right? So if you take this, here's seroplasm, which is actually a biomarker that's in plasma. It's measured for lots of different things. Here's this individual peptide. Here's transitions that are measured. And you can go and you can trigger product ion spectrum, near the apex, this peak, and you get this nice spectrum. And you can see all the Y-ion series, etc., which suggests this is a fairly abundant plasma protein. It's not that low abundant at all. And you can say, okay, well, this is our molecular species. We feel pretty comfortable with that. And then you could take this one, which is a much lower abundant protein, and you see a chromatographic peak. And here are the chromatograms for all the different product ions. And you can even buy standards, and you can confirm the retention time. You can confirm the rank order intensity of all the individual product ions. But you trigger a product ion spectrum right near this apex, this peak. And this is the product ion spectrum, and it's got all these peaks that are way, way, way more intense than your individual things you're measuring. But all the fragment ions are all there. So there's something that's present at that time, at that precursor mass, that's maybe about 50 to 100 times more intense than the peptide that we're interested in. Now, this is really the power of MSMS, right? The power of MSMS is its selectivity, is that you could have things that are a factor of 10 or 100 times more intense with the same precursor mass at the same retention time, but yet you're still able to quantify it based on product ions. We'll talk more about this later. Now, the last thing that I want to bring up is whether or not we should be putting all this effort into measuring reliable peptide areas. So there's lots of times people talk about spectrum counting and whether or not that's a quantitative approach. And we often get asked this during the course, so I've started in the last couple of years to kind of just add a couple slides right at the beginning from our perspective, because we won't be talking much about spectrum counting at all throughout this course. And so this was an early experiment we did in around about 2006, 2007. And here was a fairly simple experiment, and that is we took a human cell lysate, and we had 4 micrograms, and then half of that we added in 0.1 micrograms of E. coli, and to the other half we added to 0.8 micrograms. So if you made measurements then, the human proteins should be 1-to-1 between the two different conditions, but the E. coli protein should be basically 1-to-8. So if you had this distribution of every peptide you detected for human and E. coli, you'd see the human one should basically be, with no change, and the E. coli one should all be around this 8-fold change. But yeah, so it was basically controls, and those are 8x change. And this was, old data. This was all low-resolution mass spectrometry. I'll point this out. This was low-resolution ion trap mass spectrometry for doing peak area measurements. And we had the ability to do, spectrum counting, etc. First thing that was kind of key to kind of point out, and that is the integrated peak areas. So we had this approach, which is still actually the same peak area integration that's used in Skyline. As we call this crawdad peak area calculation, and it's still the main way which we calculate peak areas in Skyline today, with a lot of changes and improvements. Of course, it's been a long time and a lot of work. But basically, the intensity of these things, and this was a linear ion trap mass spectrometer, and the MS1 signals span, basically 4 logs of intensity that were the differences in the MS1 signals for the peptides that were detected. And if you looked at spectrum counts, though, that they only span basically 2.5 orders of magnitude of dynamic range. Okay. And then if you start to go and say, well, let's look at the E. coli signal intensities. Well, all the E. coli peptides or proteins they kind of were centered very nicely around this 8-fold change with a fairly Gaussian distribution around them. When if you look at lots of different ways of calculating spectrum counts, you notice that there's this skew to kind of underestimating things, and the distribution is much broader. If you go and look at just the signal intensities, so if you look at integrated peak area, which is this crawdad ratio, versus the intensity, and if you notice that the human proteins there was a distribution. It's fairly noisy, but it's distributed around a 0 change, and the E. coli ones are distributed around this 8x change. If you look at spectrum counting, though, it looks kind of like a mess. And that is that a lot of your spectrum counts are done with 1 or 2 spectrum counts, or 1 and 8 spectrum counts, and so it's really hard for you to get an 8-fold change with spectrum counts, because you're looking for things where you have now 10 and 80 or 100 and 800, and so those are things that are pretty hard to do based on with dynamic exclusion. Now, so what I'll point out here is stuff that's very similar to what we were bringing up earlier. While spectrum counts may not be very quantitative, it can be potentially differential. So you can find things that are different, but I wouldn't necessarily trust the fold change in the spectrum counts as being a good measure of the quantity. The other major issue, too, is that you never get enough spectrum counts on a peptide. And we already know that what is a protein. We know there's different proteoforms, etc. So it's really hard to be able to sit there and extrapolate. These are extrapolated to protein levels, right? So, which is, again, we've already discussed that may not be the best or optimal way to do things. Again, this is just something that's kind of important for people to think about, and it's not. I don't want to dwell on it much. There is definitely still usage for spectrum counts. It solves a lot of people's problems, but the reason why most people steer away from using integrated peak areas is because integrating peaks is hard. Getting good chromatography is hard. Getting nice peak shape is hard. And so that's basically a challenge. And generally, if there's one thing that works just really well for a field, that is you get a bunch of MS spectra, put it through a search engine, you get a bunch of IDs, right? And so those things, that works fairly seamless. A lot of the other tools tend to still be a challenge. But I can assure you that the quality of the quantitative data is pretty high. And so almost the entire, this entire course is going to be focused on, chromatographic peak integration. Again, so one extra point that I'll bring up is a lot of people would say, well, I don't trust spectrum counting unless it has greater than 10 MS spectra. And so, again, this is just showing that the results of the integrated peak areas with, that were not detected with less than 10 MSM a spectra versus the, spectrum counting with, and you'll still see that the data still looks quite a bit better. Okay, so in summary of this intro, and that is that immunoassays are an imperfect solution to protein quantitation problem. Don't let anybody tell you that they're a perfect solution. I think that's kind of a key critical thing. And it's a good thing, and I'm not saying that all immunoassays are bad, but I'm saying that it all depends on the antibody, and we often have no idea what the antibody is really recognizing. If you are using internal standard, add it as early as possible to account for sample preparation errors. Systematic errors do occur and need to be corrected. We talked a little bit about the fact that peptides may reflect different portions of an individual proteoform, and so high sequence coverage definitely does help and is important. It's important if you do use an isotope enrichment of the internal standard, you would like to know it, and sometimes in our lab we spend a fair amount of time actually measuring the enrichment for everything we get sent. We don't trust what the manufacturer tells us they are. 13C labeled internal standards aren't perfect. You don't just want to go and measure a one-to-one response. With 13C in particular, you definitely need to correct for the difference in the effect of the monoisotopic mass. In fact, I showed you an example where it was off by 10% just by the 9 13Cs. You should be thinking about how you're going to calculate the significance, the statistical significance before you do the study. Get help early if you're not sure. In fact, regardless, it's good to talk to people, and the more you sort of talk to people who have lots of experience in doing these sorts of things, the better off you are. And then things like, is your sample size and experimental design going to answer your questions? And the final thing is, I think, is don't tolerate experiments with an N of 1. I'll point out you'll see lots and lots of iTrack or TMT experiments. Because there's only a maximum of 10 channels, if they have 10 conditions, they'll only have one thing in each component, right? And they can't do replicates because they sample different things in the different replicates. So it's just a key component to kind of keep in mind when you're reviewing papers, etc. There's still a lot of these things that occur in the literature, and you need to say, can you actually say that difference is caused by the biological response? You can't if they don't have biological replicates. That's a kind of a key component here to think about. And I'm happy to answer any questions. It's been kind of a little quiet. I know this is kind of the first day, so please feel free to ask questions. And yes.
Can you elaborate on the future of DDA? I mean, you said that it has all its limitations, but in the past 10 years it's been the workhorse of the proteomics. Are we going to do all DIA after this? Or?
I wouldn't say so, but I also will point out too, it's kind of difficult to steer the Titanic. Once people are so used to doing things like that way, and as I pointed out, the one workflow that we have that just works pretty seamlessly is that as you collect an MSM spectrum and you stick that through a search engine, you get results. And one of the things I think will kind of come out of this is that a lot of labs haven't done, not everybody, no, not a lot. There are a lot of labs that haven't done good quality control. They haven't checked to make sure that they're getting good chromatography, that there's no systematic biases, that they're not losing response over time. With spectrum counting, these things don't matter as much, but these things become a much bigger problem when you're trying to integrate peak areas. You need to have symmetrical peaks. You need to have good spray stability. With again, DDA sampling results, that doesn't matter. And so I think basically these things are somewhat an artifact of that these experiments are hard. And the one thing that kind of comes out about it is that getting peptide IDs is easy. The problem is getting those same peptide IDs across 100 samples is hard. And so one of the things that we often try to think about, at least in our lab, and I'm not saying that this is the only way to do it, but sometimes it's better to measure a few things really well than to measure lots of things poorly. And I think that's kind of the things that we try to take to heart many times, that we don't necessarily want to measure lots of things poorly, so we spend a lot more time thinking about that. And also, I'll point out that a lot of things really changed in our lab when we really started to do lots of rigorous system suitability. Now what we end up finding is that our instruments seem to be down a lot more, or at least down. They're not really down, but they're not working in the case on a level that we would say that are optimal, or that we would want to run our most valuable samples. And when previously lots of very valuable experiments were done, and then people said, oh, it's the samples that are wrong, or this is an issue. Now we know when there's an instrument problem or a sample problem, and we feel fairly confident about that, which is something that we didn't do previously. We'll talk a lot more about this throughout the week, I think.
So what do you think about doing relative quantification instead of absolute quantification? Because
Yeah.
you cannot reproduce that data in other lab, and
Yeah.
they cannot use it for like
Yeah.
real purpose.
So, is there such thing as absolute quantification?
Yeah. Like people are misusing that data when you say that picomole per milligram of this.
So what makes something absolute?
Heavy label standard, absolutely necessary for quantification.
So you're saying someone can't do absolute with an ELISA? You go to the doctor's office, they measure a protein, they tell you the micrograms per milliliter or per deciliter of that protein.
You have to be confident in what amount you spike in to know whether you can rely on that.
But you need to spike anything in. So in the case that you're making something relative to something, right? And it all comes down to whether or not you're making it relative to something that you believe you know. It doesn't have to be an internal standard. It could be an external standard, which is what ELISAs do, right? They have an external calibration. When you do your BCA assay, people will often go, I'm going to make it better relative to albumin. Albumin is not a good calibrant often for a lot of whole cell lysates, but that's what people do, right? And anyone can go buy albumin and make their measurements relative to albumin, right? So that's a fairly easy thing to do. The problem is if I go buy a peptide from New England Peptide, and you guys go buy that same peptide from Thermo Fisher, are they going to be the same level, the same amount? No, probably not. And so that's a big problem we have. And I'm not sure how many people realize this, but we actually have an entire institute at the United States called NIST. And their entire thing was basically to kind of come up with standards that we make everything relative to. If you're trying to think about what's time made relative to, well, it's Greenwich Mean Standard Time, and often the atomic clock is in Colorado, right? That people tend to set as far as the time goes, and as far as a meter stick, we have a fairly good measure of what that meter stick is, and we can make our other measurements to how many meters something are because of meter stick. The problem is that we don't necessarily have good standards to measure things relative to. Now, there are some good clinical assays where they now have some really good reference material. Right? So they'll have like a plasma where they say, look, thyroid globulin is this level. Or they'll have a thyroid globulin standard at a really well. And it doesn't actually really matter what that level is as long as everybody can make their measurements relative to that. And it's the same thing. So carbon 12. Carbon's 12, not because it's 12, it's because we said it was 12. So again, it really just matters, okay, I'm going to take this level, I'm going to call it X. And then everybody else measures their thing relative to this thing that I'm making my measurement to. That's fine. So my personal opinion, and I know Andy Hoofnagel will talk about this a little bit too during the course, and that is that there is no such thing as absolute quant. Everything's relative quant, and it just depends on what you make your measurements relative to. You definitely don't need a stabilized labeled internal standard in order to make it. In fact, there's a lot of confusion within the proteomics field that an internal standard is an internal standard. It's to correct for reduce the variance and improve the precision. And then there's calibrants, and sometimes people use the same thing to do both calibration, and that's an internal single point calibrator. And so a lot of times you can do an external single point calibrator. Now there may be issues with precision, et cetera, that could affect it. But I think some of these things will get discussed during the course. If not, I'm definitely happy to kind of chat over it. There'll be lots of times to kind of talk, I think, throughout the week. Yeah.
I would just say that, there is still a very important distinction that I sort of had to get my head around for absolute, what is called absolute quant. It's like, it produces a measurement that you could get, that a doctor could give to somebody, or that you could set a standard and say, like, a patient between this range and that range, has this risk of this disease, which you can't do with a relative measurement like a peak area, or you can't just say.
But those measurements are wrong. So Andy will I'm sure he'll present a case of one patient going to 3 hospitals in the Seattle area, getting an ELISA done with an absolute measurement, and them getting 3 different measurements of which change their
Well,
treatment
it's a
options.
poorly calibrated absolute measurement, but it's
Yeah.
still,
Right.
the intent
Yeah.
is that you can say, okay, mass spectrometry doesn't allow us to get as well calibrated absolute measurements as the meter stick. Like a meter stick, you say, is a relative measurement. The attempt is still to produce an absolute measurement. You could say it's well calibrated, it's poorly calibrated, but the attempt is still to produce a well calibrated measurement.
I think what comes down to just having reference materials. So one of the great things in plasma is Golden West Biologics is a fantastic resource. They have pooled lots that have 400 patients and things too. And every experiment that we've done here and that the hospital here has done, they've made their measurements relative to that, and it's easy for someone else to go buy that same lot and make measurements relative to it. And whether it's metabolites or proteins, etc. Now, when we do work with the Rabinovich lab here, one of the things we've done is we do a lot of heart work. So we've taken a ton of their hearts and made a big pool, and we've got an enormous amount in it that's fractionated out. Every time we have a batch, we take one of those things and make the measurements relative to it. And we now even have people who are asking us for some of that measurement so that they can make their measurements relative to that too. So again, it's calibrated, it's just right now it's X, right?
But so I think using Andy's example is a little bit like saying we can't have absolute time, because if I ask everybody who's got a watch in this room what time it is, I don't get the exact same answer.
Yeah.
I mean, it's still, it's still possible to set these things up, and there is a meaning to give me a reading of time.
Yeah.
And it's different from a peak area.
Yeah, and all I'm saying is that it all depends what you make that thing relative to. So, and it doesn't really matter, because we know that a meter stick or a yardstick, they're different values, but it doesn't, and you can calibrate them to different things. It could be in meters or yards or miles or kilometers but you do need to know what is it you're scaling it towards and what the comparison is. And all I'm just trying to say is that every measurement's relative to. The problem is it's really hard in any protein measurement to have something that's known. That's a tough thing, I think, for quantitative scientists. So when I was doing isopressure mass spectrometry, I'll give another example. Everybody measures 18O content in water using standard mean ocean water. There's no shortage of standard mean ocean water, right? So, and that 18O content is the same across all standard mean ocean water. So if you're making it relative to standard mean ocean water, it's really straightforward to do that. Now, 13C values are much harder, right? Because 13Cs, they change in carbonate rocks, they change in the body. They depend on whether you have like a maple trees versus corn syrup. They all have slightly different 13C values. So they used to always make this measurement relative to a fossil called PDB, PD belemite. And PD belemite is a fossil that was only present in the world at a very narrow point in time when the 13C atmospheric content was very constant. So it actually has, no matter where they find that fossil, they have the same 13C value, right? So the world makes their values relative to 13C PDB. It's a delta 13C PDB. And so the problem is now they can't find any more of this fossil, right? So, but NIST made standards relative to PDB, and people then have their own working standards they make relative to that NIST standard relative to PDB. But again, everything's relative, and it just happens to be that PDB happened to be a good measure, and now it turns out that NIST tends to be a measure relative to PDB that everyone sort of calibrates relative to. But it's But the problem that we're going to have, and we're going to face for a while, I think, is what is the known value. And so I know for thyroglobulin has been a huge success in the clinical laboratory, and a big component of making it so that the measurements come back across many different labs, that NIST has a really good reference standards that everybody calibrates relative to. And stabilized label and peptides are going to be a hard thing to do. That's all I'm trying to say. So I agree that they're calibrating to something, and definitely if you can calibrate to something and to give that something to somebody else that they can calibrate to, is even better. So we have a case now where we're trying to do formalin fixed paraffin embedded measurements. And what we've made is these tissue blocks with a known, what we call a known quantity. It's just a fixed homogeneous quantity of a protein in a tissue block. And every time we make a measurement, we take a section off that, we make a measurement relative to that. Anybody who wants that block, will send them a section, right? And they can make the measurements relative to that too. But, and when that block runs out, we're going to have to make a new block. We're going to have to make a new block as known quantity relative to what the old block was, right? Which is like the whole PDB problem again. Right? I'm not saying that these things are impossible. I'm just saying we haven't thought about them yet because they're still fairly new. And they're not just limited to mass spectrometry. They're also limited to like doing your BCA assays. If you use IgGs as your calibrant as opposed to albumin, or if you use plasma as your calibrant relative to albumin, or if you use a cell lysate relative to, it's just, you'll get different results. And it all comes down to what you calibrate it to, and it's a problem. But as long as you know that that's a problem, that's fine. And you know the limitations. I think that's the kind of the key component. But I think Brennan's right that having a calibrated value is important. The problem is being able to share enough of that reference so that people can make the measurements relative to. And some fields have done better jobs of that than others. Yeah.
Maybe one comment to your problem that you explained that you cannot expect a 1-to-1 ratio when you work with 100% related standard. And my comment to that is this issue that you will have maybe 10% difference is especially true, or is true when you use only the monoisotopic peak for quantification.
Yeah.
But a lot of tools don't do this, so a lot of tools use the full isotope, and in the moment when you use also the second and third isotope, it's much less efficient.
Yep. Yeah, actually, I have an animation set slide that basically shows that the area of all. So you basically have what's called mass balance. So the sum of the isotope distribution is actually the same, but the monoisotopic mass isn't. So there are different tools that you take different approaches for doing these things. When you include more isotopes, you include your bigger chance of interference and other problems, but it does reduce some of these systematic biases. And things like SRM often will just have a narrow isolation window around the monoisotopic mass, but sometimes it includes other isotopes too. So it's important, I think.